So the last section of this piece is probably the most interesting because we're going to be talking about mutations. So mutations are any change to the genetic information of a cell or a virus. Now our focus for this section is on small scale mutations that involve only a few nucleotides, like one to a few small amounts. So point mutations are the chemical changes in just one nucleotide pair of a gene. So what this can cause is if we change one nucleotide in a DNA template strand, that could lead to the production of an incorrect protein. And if that were to occur in a gamete cell, that could even be transmitted to the offspring. So one example of a point mutation is sickle cell anemia, which you've probably heard about. The normal red blood cell looks like this, but the sickle cell blood cell looks like this. And this is due to a point mutation in the gene encoding the beta globin protein or polypeptide of hemoglobin. So this leads to the production of an abnormal protein, and whenever you have a change in the protein, that can sometimes result in a different phenotype or look of how the gene is expressed physically. So the typical normal beta globin protein looks like CTC, but in the mutant beta globin protein, you'll see we get CA instead of CT. So that means that the opposite DNA strand is going to give us a GT instead of a GA. And then whereas the original mRNA would correspond to GAG, the mutant mRNA is going to be GUG. And so whereas the normal hemoglobin is includes the amino acid I believe this is glutamic acid. Instead, it's going to change to valine because this codon codes for a different amino acid, and that's what results in a different phenotype. So remember, it starts with the genes to mRNA to protein, and the protein is what we see expressed. So there are kind of two different point mutations that we're going to be talking about. One is known as a single nucleotide pair substitution, and the other are nucleotide pair insertions or deletions. So a nucleotide pair substitution is just like what it sounds like. It's when one nucleotide and its partner are replaced with another pair of nucleotides. And this can result in a few different scenarios. So if you remember back to the codons in the transcription lecture, there are several codons that code for each amino acid. In most cases, each amino acid has at least 2 codons that code for it. And some of them even have 3 or 4. So in some cases, when this substitution occurs, they end up coding for the same amino acid, even though it's a different codon. And so this would, as you can imagine, have no effect on the amino acid that's produced because it's the same one. And so we call these silent mutations because even though this is a mutation, looking at the actual protein, you probably would have no idea that there was anything wrong. So this would be an example of a silent nucleotide pair substitution. So in this one, instead of a G right up here, it changed to an A, and then the pair would be T in the template strand. In the mRNA, we're going to get a U, but this group GGU and GGC both code for the amino acid glycine. So you can notice the amino acid chain is not actually going to change because these codons both code for the same amino acid. On the other hand, with something known as a missense mutation, this is a substitution that changes one amino acid to another. And so in this case, we have GGC, but now when this gets changed at the top to a T, we have AGC, and this AGC codon codes for the amino acid serine. So you can notice that this is very different. Our missense mutations, some may not have a major effect on the protein as a whole, since it only changes one of the amino acids. If the amino acid functions relatively the same, then the protein might be relatively unaffected for a missense mutation. On the other hand, if the amino acid changes in a critical area of the protein, it can majorly impact the activity and function of a protein. So missense mutations sometimes are not harmful, sometimes they are. The most dangerous mutation, however, is known as the nonsense mutation, and this can actually cause a problem when they change the codon for an amino acid that's functional, such as lysine, to a stop codon like UAG. In this case, what happens is translation is terminated early, and almost all of these situations in a nonsense mutation lead to a non-functional protein, because as you notice, this protein had quite a few amino acids that were a part of it, but when this mutation occurred, it stopped at methionine. So really nothing happened after that point, and this protein thus becomes unusable and non-functional. Even more dangerous than these nucleotide nucleotide substitutions are insertions or deletions, and these are situations where we actually get additions or losses of nucleotide pairs in a gene, and these can be disastrous because if they're not in a group of 3, then you can get a very, very big problem because the whole frame, the whole reading frame of the mRNA is messed up. And this can have a huge disastrous effect on the expression of the gene. So we call these a frameshift mutation because these insertions and deletions completely affect the way that the gene will be expressed, and I'll show you why. So one thing that could happen here is you can see in the original protein we went, or in the original DNA we went TACT, and here we have TACA. But notice it continues on TTC, TTC, so we have this addition here. And what happened here is we ended up getting a UAA, which is a stop codon. So this caused immediate nonsense, just like in that nucleotide substitution, it just stopped. But perhaps even more dangerous is here when we have a deletion, and this frame shift took out this amino acid, and because this is a non-overlapping code, if one of those if one of those nucleotide bases goes missing, the whole code gets messed up. So now that this is taken out, instead of having a UUU, we have a UUG, which codes for a different amino acid. But this G, you can see, moved over because that U was missing. And so everything else shifts, hence the name frame shift, and the whole reading frame shifts. So not only does this guy get messed up, but so does this guy, because it's now no longer GGC, that G came over here, it's now GCU. And the next one is going to get messed up. Notice this one was originally a stop codon, but now we don't know what it's going to be. So the frame shift mutations are very, very dangerous and can cause a lot of problems. This, just as a reminder, this is some more information about the nucleotide substitutions. This is what I've already mentioned, but if you wanted to copy down the notes, you can pause here and do that. We talked about insertions and deletions. And the last question a lot of people have is, how do these mutations actually occur? So one type of mutation cause is just known as spontaneous mutations. And this is any mutation that happens on its own during DNA replication, recombination, or repair. So kind of like what we talked about at the very beginning of this unit when DNA copies itself. A lot of times it checks itself and proofreads before it finishes off, but if it misses anything, that would be a spontaneous mutation. Then you may have also heard about substances that are mutagens, and these are physical or chemical agents that can cause mutations. So one example you might have heard of is X-rays. The reason why you have to wear that like giant lead cape when you get your x-rays taken at the dentist is to prevent mutations from occurring in your cells that could maybe cause cancer. So those x-rays are mutagens. They can mutate your cells and cause a lot of errors. So finally, coming back to what we started with at the beginning of this section, when we talk about what is a gene, there's a lot of different ways that we're going to talk about a gene, but for us, probably the best definition would be that a gene can be defined as a region of DNA that can be expressed to produce a final functional product, either as a polypeptide or a protein or an RNA molecule. However, in the last section, or the next section that we're going to be doing, we're going to be talking about genotype versus phenotype. And phenotype is the physical expression of a gene. So for example, I have blue eyes because that is my eye gene's phenotype. And in that case, it's more useful to think of the gene as the protein, because when the protein is expressed, that's going to give us a particular phenotype. So just as kind of a review of this whole section, remember we always start with DNA. That's what's the code for our genetic information. Through transcription, we get pre-mRNA in eukaryotes or mRNA in prokaryotes. That mRNA is processed further in eukaryotes. In prokaryotes, it goes straight to the cytoplasm. In eukaryotes, it's processed to become mRNA and then moves to the cytoplasm for translation. And in that translation process, we end up, with the help of ribosomes, forming a polypeptide or a protein. So that really sums up the way we get from a gene to a protein.